On behalf of the Vice Chancellor and Principal, Professor Nana Poku, it gives me great pleasure to welcome you to the inaugural lecture of Professor Augustine Gubba. The Vice Chancellor conveys his congratulations and best wishes to Professor Augustine Gubba. The Registrar, Dr. Catherine Cleland, conveys her apologies and congratulates Professor Gubba on his promotion and thanks him for his contribution to the School of Agriculture, Earth, and Environmental Science. I would like to acknowledge the following guests this afternoon: members of Council, members of the Executive Management Committee, members of Senate, our inaugurant this afternoon, Professor Augustine Gubba, family and friends of Professor Gubba, academics and professional staff, students, alumni, and distinguished guests. A special welcome to our guests from universities and organizations within South Africa, the African continent, and across the globe. Inaugural lectures form part of the university's public lecture series and may only be presented by newly appointed full professors who have been appointed in academic schools and centers. Inaugural lectures present an opportunity for showcasing the exciting and groundbreaking teaching and research being carried out by professors in our university. Each lecture represents a significant milestone in an academic's career, providing official recognition of their promotion or appointment to full professorship. These lectures are furthermore an ideal opportunity for new professors to introduce themselves and to present an overview of their own contribution to their field to academic peers, students, and research collaborators. Inaugural lectures are also a platform for celebrating a professor's academic achievements with his or her family, friends, mentors, and colleagues. Distinguished guests, it is my pleasure to now introduce the Dean and Head of the School of Math, Stats, and Computer Sciences, Professor Celestina Viriri, who will now formally introduce the inaugurant, Professor Augustine Gubba. Over to you, Professor Viriri.
Thank you so much, Professor Kopenali, College Dean of Research, and all invited guests, Professor Gubba and your family and all your friends, as well as university staff present here, and the registry personnel for organizing this. Professor Augustine Gubba is a full professor in the discipline of plant pathology in the College of Agriculture, Engineering and Science at the University of KwaZulu-Natal, Pietermaritzburg campus in South Africa. Professor Gubba received his BSc Honours Crop Science degree from the University of Zimbabwe in 1984. After graduating, Professor Gubba began his career as a curriculum development officer in the training department of Agricultural Extension Services in the then Ministry of Agriculture in Zimbabwe. In 1986, he moved to the Plant Protection Research Institute in the Department of Research and Specialist Services in the same ministry. Here, his duties involved working in the Plant Disease Diagnostic Clinic and conducting research in plant pathology of field and ornamental crops. In 1987, he received a British Council scholarship to study for an MSc in Applied Plant Sciences at Way College, University of London. The degree was awarded in 1988. After completing his MSc, he went back to his position at the Plant Disease Diagnostic Clinic. In 1994, he was awarded a Rockefeller Foundation scholarship to study for a PhD in plant pathology in Cornell University, and the degree was awarded in year 2000. After working as a postdoctoral associate at the Glansoff Laboratory at the Geneva Experimental Station, part of the Cornell University from January to November year 2000, Professor Gubba joined the discipline of plant pathology at the then University of Natal at the end of 2000. As a senior lecturer, he was promoted to associate professor in 2017 and ascended to the position of full professor in March 2023. Professor Gubba's research area is in plant virology, The main thrust of his research is the identification and characterization of viruses infecting important food crops, with the aim of devising effective and sustainable strategies to control and manage the disease they cause. He has successfully completed the supervision of 8 doctoral and 15 master students. Professor Gubba has published over 60 research articles in Scopus and ISI indexed journals published by Springer, Taylor and Francis, Elsevier, among others. He has also contributed chapters in 3 books. He is currently supervising 2 PhD students. Professor Gubba is responsible for teaching 3 exit level modules at the 3rd year level and 1 at the 4th year level. The modules are Introduction to Virus, Biotechnology for Plant Pathologists, and Advanced Topic in Virology. Professor Gubba has examined a number of master's and PhD theses from several universities, including the University of Witwatersrand, Stellenbosch University, University of the West Indies, and Northwest University. He has reviewed research papers for publication in a number of journals in his area of expertise. Professor Gubba has reviewed research projects for funding by the USA National Science Foundation and the National Research Foundation, NRF of South Africa. Professor Gubba has an NRF C2 rated scientist since 2017. He was a recipient of the Distinguished Teacher Award in the School of Agriculture, Earth and Environmental Sciences in 2014. From March to November 2023, Professor Gubba was a Fulbright visiting research fellow working jointly with the United States Department of Agriculture, Vegetable Research Laboratory, and Clemson University's Coastal Research and Educational Center based in Charleston, South Carolina. The fellowship gave Professor Gubba an opportunity to hone his skills in CRISPR and Cas technology, which will be part of his research focus going forward. Professor Gubba is a commit member of the International Committee of Plant Virus Epidemiology and is one of the 2 African representatives on that committee. His expertise has led to him being appointed as a genetically modified organisms officer for the South African government, reviewing applications for GMO product registrations and research facility registrations. He is also a technical specialist for the South African National Accreditation Systems, SANSA, assessing laboratories seeking accreditation for various techniques in molecular biology and plant pathology research. He was vice president of the Southern African Society for Plant Pathology from 2015 to 2019. Professor Gubba has been used as an expert witness in court cases that needed technical plant virology input. I now invite the inaugurant Professor Augustine Gubba to present his lecture.
Thank you, Professor Neil. Thank you, Professor Viriri. I'll start. Let me share my screen. There we are. I want to start by greeting all in the audience. Thanks for taking time off your busy schedules to listen to my inaugural lecture. Before I start, I want to dedicate this lecture to my parents, my late father, John Bebura Gubba, and my mother, Irene Gubba, who both as educators instilled in me the need to appreciate the value of education in life. I will forever be indebted to them both. My lecture is an encapsulation of the journey I've traversed throughout my career. I will use the lecture to answer the following questions. Where did the journey start? How did we end up here? What are the highlights along the way? It will become clear that I've used viruses, but specifically plant viruses, as the preferred mode of transport to traverse this journey. I'm well aware of the varied backgrounds of people in the audience, so I'll do my best to keep the presentation very simple without losing meaning. I'll start by addressing the size issue, which is part of the title. So the question that one asks is, does size really matter? Viruses are among the smallest microbes and are the most abundant biological entities. They are measured in nanometers, which is 10 to the minus 9 meter, power 9 meters. And this means basically that they are invisible and they can only be viewed using an electron microscope after being magnified several thousand-fold. So what does this mean in real terms? 500 million rhinoviruses, this is the virus that causes common cold, could fit on the head of a pin, 500 million. That is how small viruses are. Viruses are molecular parasites that only come alive in an appropriate host from where they can unleash a lot of havoc by commandeering the infected host cells to produce their component parts. And this is what results in disease. So let not their small size fool you. Viruses are a clear case of dynamite coming in very small packages. Structurally, they are very simple. They are made up mostly of genetic material, which can either be DNA or RNA, which is surrounded by a protein coat. Some viruses have envelopes. These envelopes are actually obtained from the host that they infect. So for the viruses that I'll be talking about, which are plant-infecting viruses, these are the shapes that one sees when he looks at them under the electron microscope. So they appear in different shapes and sizes. And now I want to address the other part of the title, which talks about the power of the virus punch. And the term virus to most in the audience will refer to those viruses that cause human and animal diseases. The debilitating effects of polio and smallpox, the devastating decades-long effects of HIV, the deadly impact of cervical cancer, the recent world lockdown due to COVID-19, the current egg shortage in South Africa due to avian influenza, and the irritating effects of common cold are a few examples of the impact of viruses, an illustration of the power of their punch. There is also the other side of the story, which is seldom told, where viruses have devastating effects on the crops we grow for our food, making them a serious threat to food security. This is my office. This is what I do, working with such viruses. So welcome to my world. I'm going to use two examples to illustrate the power of the punch of plant-infecting viruses. The first example is a disease of maize called maize lethal necrotic disease. This disease is a result of the synergistic interaction between two viruses. And as you can see from this slide, a farmer with that crop is going to harvest nothing, zero yield. The crop has been totally destroyed. Another example that I'm going to use is that of papaya ring spot infecting pawpaw plants. And this, what you see there is a once productive pawpaw farm. But after a while, because of the papaya ring spot virus, that farm was abandoned due to lack of productivity. So how do viruses impact crops? There are different ways in which they do this. There's reduction in growth, reduction in vigor, reduction in quality or market value of the produce. And there are also costs of attempting to maintain the crop health. And all this translates into losses, which is worth billions of dollars. With that said, it is clear that viruses pack a very powerful punch in spite of their small size. In fact, I dare say they're always punching way above their collective weight. And as for the symptom that they cause on the plant that they infect, the most common symptom, you see it on leaves as yellowing or discoloration. Sometimes you can also get plants being stunted, stem pitting in some cases, and sometimes the symptoms are exhibited on the fruits, making them unmarketable. When it comes to the control of viral diseases, it's not easy. This is due to the fact that viruses are always evolving, which results in mutations in their genomes. They are like a moving target. So it's very difficult to pin them down. Be that as it may, there are a number of recommended strategies that can be used to control or manage viral diseases, and I've listed them there. You remove infected plants, you use virus-free planting material, breeding for resistance, and cross-protection, which is a form of vaccination by using a mild form of the virus. But the last method, that is what I've highlighted in bold there, biotechnology, this has only been come into use in the recent few years. And people use it only when all else fails. In other words, when the other four things don't work, people resort to biotechnology methods. I want you to keep this at the back of your mind, because the research that we do is based on biotechnology methods to control virus diseases. So now I'll go back to the journey. Where did it begin? Some of it was already mentioned in the introductory remarks, but the seed of interest in plant virology in me was planted during my time studying for the BSc degree, honors degree at University of Zimbabwe. I took a liking to plant pathology thanks to the excellent teaching of the late Desiree Cole. And the first viruses that I encountered during that time were groundnut rosette virus and maize streak virus. For those of you who are familiar with the crop science building at the University of Zimbabwe, at the back of the building, the experimental plots. So it was on these experimental plots that Desiree Cole took us as students to show us the effect of viruses. And the top one is showing plants, maize plants with maize streak. And then the bottom one is showing a healthy groundnut next to one infected with groundnut rosette virus. The impact is obvious. So I became fascinated by these invisible microbes, which had profound effects on plant growth. It stuck with me. I'll hasten to add that at this time, it was all theoretical virology. We had no equipment or budget to do virology research. They were really frustrating years. I think many budding virologists in the developing world can easily relate to this. That is why when I teach my students, I always impress on them that they should appreciate what they have, because at this stage, they can do anything they want, things that some of us used to dream of when we were undergraduates. So going to the early years of my work, my first job was working for the Agriculture Extension Service. I was part of the training department responsible for curriculum development. And at that time, Zimbabwe was world-renowned for its agriculture productivity. So I was part of the extension service, which was training the extension officers. And here I worked under the tutelage of Jeff Mutimba. And I think this is when the teaching bug must have beat during that time. So within a year, I had to switch to the research space because I wanted to grow. This I was encouraged to do by my good friend, Professor Paramafongoya. So the research obviously offered better opportunities to grow and for me to start working on the invisible pathogens I had encountered at varsity. This was an excellent opportunity. So a year after the switch, I was attending my first international conference where I was presenting a poster on barley yellow dwarf virus, which is a cereal virus. This conference was held in the city of Trieste in Italy. I did it in style. CIMIT, who were the sponsored, paid for first-class travel on British Airways. This was my first time on a plane. The culture shock is a story for another day. Later on, I participated in a survey of cowpea viruses as part of a group of African virologists in a project that was coordinated by IITA, Ibadan, Nigeria. Several trips were made to Ibadan on this project. My career as a plant virologist was now beginning to take shape, and I must admit, I was enjoying the traveling. For my MSc project, White College, I worked on a virus, cowpea aphid bone mosaic virus, under the supervision of Keith Bayliss. During this time, I also used to help teaching virology in biological sciences department at the University of Zimbabwe. And I also spent time at CIAT in Colombia, working on bean viruses in the lab of Francisco Morales. I mean, at that time, it was an interesting time to be in Colombia. The drug cartels were running the show, and the name Pablo Escobar comes to mind. So early publications of that work are listed here. And the first one to me is very interesting because I was very proud to be associated with people who were the big hitters in virology at that time. George Thotapili, Richard Hamilton, Russell, Shonika. So we were, our names were next to them in this publication, and that made me very proud. Moving on to my PhD research. So in the early 90s, the University of Zimbabwe and Cornell signed a cooperation training agreement in horticulture. And the leading virologist at Cornell, Dennis Gonzalez, he came to Zimbabwe as part of this program, and I hosted him. The reason I hosted him was I was the only one who was working on virology in Zimbabwe at that time. So he came to me, I was shadowing him for a few days, the time that he was in Zimbabwe. I took full advantage and used the opportunity to talk to him about my aspirations to be a fully fledged plant virologist. That year, I was awarded a Rockefeller Foundation Fellowship, which I used to apply for graduate school at Cornell. And then it allowed me to link up with Dennis for my PhD research. And as they say, the rest is history. So now in joining that Gonzalez lab was at a time of the early years of genetic engineering. That is when people were beginning to develop GMOs. I became part of a very big lab. I know at one time, there were 23 of us, PhD students and postdocs, all foreign students. Can you imagine if a professor here can do that? You have 23 people in your lab, all being foreigners. Okay, at that time, the concept of pathogen-derived resistance had just been described. And according to this concept, if you take a gene from a virus, a small piece of the genome of the virus, and you transfer it into a plant, in a way vaccinating the plant, that plant will show resistance to the virus. So this became the basis of developing transgenic plants with virus resistance, their GMOs. And the Gonzalez lab is famous for developing virus-resistant transgenic papaya for Hawaii, which became the selling point of the new technology. So just to let you in on the process of developing GMOs with virus resistance, what exactly do we do? So you start with the pathogen. Here it's labeled as bacteria, but replace that with the virus. So you go to the virus and take a small part of the genome, the RNA or the DNA. You clone that, you make many copies of, and then you design the gene. So that process of designing the gene is called genetic engineering. There's a lot that is involved. So the moniker that we have gained as people who do this kind of work, people refer to us as genetic engineers because of that process. And some condescendingly refer to us as gene jockeys. But anyway, so once you design that gene, you come up with a gene construct that you want to put into the plant. It's not a simple process of injecting into the plant. There's a prescribed way of doing it. You are going to use the gene construct to inoculate small pieces of the plant that you want to transform, and then via tissue culture, you regenerate that plant. And that plant will have the gene of interest that you obtained from the virus. So in a short, brief explanation, that is how we develop these transgenic plants. So as for my PhD project, I worked on a very important virus called tomato spotted wilt virus. It is considered among the top 10 viruses that infect crops based on the damage that it causes on several crops. Losses that it causes run into billions of dollars. And you can see the different type of symptoms that it causes on different hosts. It has got a very wide host range. And farmers have got very few options available to manage the diseases that this virus causes. And this is where now the development of transgenic plants with virus with resistance becomes an option. So this was the focus of my PhD project. So I went to the virus, which looks like this. It is one of those few plant-infecting viruses that has got an envelope, the envelope I referred to earlier on. So I focused on the gene here, the nucleocapsid gene. I took a small piece of it, did my genetic engineering, and then went to the tomato that I was transforming, inoculated small pieces of the tomato, and via tissue culture, generated these tomato plants, which now have got the virus, the gene from the virus. So there are several tests that one has to do to test whether the gene is present or not. But the most important test is when you inoculate the plants with the virus from which you obtain the gene to show that it is resistant. And this slide here clearly demonstrates that. These two lines here are transgenic plants. When inoculated with the virus, they show resistance. According to the concept of pathogen-derived resistance, it makes sense. And then these other two lines, when inoculated, because they are non-transgenic, they are going to be killed by the virus. So this is showing effectiveness of the virus. A key publication that came from that work was when we published in Molecular Breeding. We combined transgenic and natural resistance to obtain broad resistance to different viruses. That is the group to which TSWV belongs. So you can see right in those early years, the concept of gene pyramiding, we actually were part of the conversation early by that year 2000. It's now very commonly used in the lexicon of GMOs, the concept of gene pyramiding, but we already were working on it by 2002. So teaching virology. Teaching is one of my major responsibilities here at UKZN. And before I go into that, I want to address how I ended up at University of Natal. So while doing my postdoc, after completing my PhD, a fellow student, Frank Wong, alerted me to a plant virologist position which had been advertised in the Journal of Science at the University of Natal. I took a shot and sent in my application. This led to a 3 a.m. interview, 3 a.m. USA time, because of the time difference. I think it was 9 a.m. here in South Africa. So now the panel was a powerful panel, and the big professors were there. Professor Mark Lane, Professor Mark Wallace, Professor Mike Savage. I was grilled, but when everything was said and done, I got the position, and this is how I ended up at University of Natal. So when it comes to teaching, it has already been mentioned, I'm responsible for these exit module level modules which are specific to virology. And some of the students of these modules have found careers in plant and medical virology. I'm going to focus on the medical virology ones because they are now in the HIV program, and by their own admission, they tell me that their career choice was actually influenced by that time I spent with them, teaching them introduction to viruses. And one of them, who has become a colleague and friend, is a member of staff in the HIV program, Dr. Paradise Madlala. He is never forgotten where he came from. He comes back to me with all the latest information on HIV, and sometimes when he's not busy, he actually teaches that part of the module for me. And for my efforts, I was awarded a Distinguished Teacher Award in my teaching efforts. I was awarded a Distinguished Teacher Award in 2014. So now I'm going to focus on my research highlights while at UKZN. As already mentioned, my research focuses on the identification and characterization of viruses infecting fruit and vegetable crops with the ultimate aim of developing effective and sustainable control management of the diseases that they cause. So while my research program was taking shape, my PhD supervisor, who was now director of USDA in Hawaii, contacted me to get involved in a project to carry out a survey of papaya ring spot virus in the Great Lakes region. He said to me, there's a very big likelihood that we're going to be funded. So please, can you assist? This actually was following reports made by my good friend Joseph Ndunguru, that the virus was prevalent in the region. So I gladly took up the challenge. We covered the length and breadth of these 3 countries, collecting samples from smallholder farmers' fields. But to cut a long story short, this USAID ended up not funding the project. However, something good came out of it. I got my first sabbatical funded by this project, where I spent time in my PhD supervisor's lab in USDA, now in Hawaii. And here we were studying the GMOs, the papaya GMOs, with a group of other African scientists, and also working on the samples that we had collected in the Great Lakes region. The beauty about being in Hawaii is you can actually see the impact of the GMO technology. On your left is a time when people were abandoning their orchards because of the virus. But the moment the technology came in, people can grow papaya wherever they want. So the technology is very effective, there is no doubt. So now for the next few slides, I'm going to focus on the research that the students I have supervised and co-supervised have done. The first one is on transgenic sweet potato, which was the work done by Dr. Bernice Sif-Prasad. You can see her when she graduated with me there. And this one was a special PhD because it was my first. So I'm very proud of it. So we focus on sweet potato because we know it's a very important food crop. But the problem with sweet potato is it is vegetatively propagated. That is, you grow it from cuttings. And that makes it very prone to virus infection. And the bad thing is infected plants don't show symptoms. They are symptomless. So there is a virus disease called sweet potato virus disease. It's actually caused by the interaction between 2 viruses, sweet potato feather remotal virus and sweet potato chlorotic stand virus. And when that virus hits, a farmer can lose between 80 to 90% of their yield. I mean, there's yield reduction of 80 to 90%. Again, illustrating the power of the virus punch. So this is a slide which is showing you on the left, this is a healthy potato, sweet potato with nice tubers, and you get good yield. Okay, and on the right is an infected plant. Look at how small the tubers are. Obviously, that will impact on the yield. So what we did with with Dr. Sif-Prasad was to go on a survey, go out and look at all the sweet potato growing areas, collect samples and test them in the lab for which viruses are prevalent. Okay, so we drove around. I was her driver. She didn't have a license. And when she analyzed the samples, 4 viruses were always occurring wherever the crop were grown. And I have referred to them here with their acronyms. Please don't worry about that. The first 2 are the ones that cause sweet potato virus disease. So now, first with that, how do you use biotechnology to develop a sweet potato with virus resistance? The way to do it is to go to each of those viruses, take a small piece of each of them, join together using genetic engineering techniques. So with that construct, which is carrying all the 4 pieces of all the 4 viruses, This is what you then use to transform sweet potato. So this is what Dr. Sif Prasad did in her work, tissue culture, until she regenerated the plants. So now the acid test is, are the plants resistant? You have to inoculate them. And this was done. And this is, the results are shown in this slide. On the right, you can see that plant which has got many leaves. This is the transgenic plant. It was inoculated with all 4 viruses. No symptoms, growing nicely. The other one, non-transgenic on the left. You inoculate them with the same, inoculated with the same viruses, less foliage. It's actually reacting to the infection. So it's susceptible. This is resistant. So clear demonstration of the effectiveness of this GMO technology. So a few publications came from Dr. Sif Prasad's work, and I've just listed a few of them in transgenic research, South African Journal of Plant and Soil, and crop protection. Moving on to the next project. This was where we focused on producing transgenic cucurbits. So we went out on a survey to find out which are the most important viruses, and we came out with 3: watermelon mosaic virus, zucchini yellow mosaic virus, and zucchini shoestring virus. So Dr. Ibaba used his genetic engineering skills, joined the pieces of those 3 viruses together and used them to transform the cucurbit. And this slide here is showing you the impact of the viruses on that small scale farmer's field. And as you can see, you can understand why the supermarket chain rejected his produce outright. Again, illustrating the power of the punch of these viruses. So after Dr. Ibaba had done his work, the acid test again is now to inoculate, and the results are very clear here. These are in the middle is your transgenic plant inoculated with 3 viruses, growing normally, no symptoms. It actually grows just as good as the negative control, which is not been inoculated. But if you look at that plant, which is not transgenic, you inoculate it with the 3 viruses, it's dead. Again, showing the effectiveness of this technology. And so a key publication from this work was one entitled Use of Chimeric Transgene Construct to Confer Broad Resistance in Zucchini Plants Against Cucurbit Infecting Potiviruses Occurring in KwaZulu-Natal. This was in the South African Journal of Plant and Soil. And as an offshoot of Dr. Ibaba's work, we actually discovered 2 new virus species, which we named here. One of them, purple aphid-borne yellows virus. The other one is zucchini shoestring virus. These viruses were accepted by the International Committee on Taxonomy of Viruses. So this was a major significant contribution to the body of knowledge on viruses by our group. And then we also dabbled with climate change and plant virology. This was the work of Dr. Moodley, Vanessa Moodley. Here we're dealing with whiteflies. Whiteflies are an efficient vector of many economically important viruses. And recently, the whitefly population has been shown to be rising. It's been on the rise due to 2 main reasons: climate change, and there's misuse of chemicals, which has led to the insect developing resistance to the chemicals that are used to control them. So Dr. Moodley undertook countrywide surveys on tomato and pepper producing farms and also greenhouses, collected samples, brought them to the lab for analysis. And when these plants the samples were analyzed, we picked up 3 viruses. Tomato torredovirus. This virus was being reported for the first time in South Africa due to this research. The others, tomato chlorosis virus and tomato chlorotic stunt virus, were already present in South Africa. But our major contribution here was to discover this virus for the first time in South Africa, tomato torredovirus. And Dr. Moodley published quite a few papers from his work in different journals, viruses, PLOS One, crop protection, and plant disease. Moving on to the next project, Indaba captures stories about graduates. And when I saw this heading, going bananas for academia. I said, oh, I think Sally Frost and her team have outdone themselves this time. This was a very catchy title, which was describing the work of Dr. Gubba. Dr. Gubba was working on bananas, hence the title of that article in Indaba. So just a brief remarks on the banana industry in South Africa. In 2021, South Africa exported 13.6 million worth of bananas, making it the 49th largest exporter of bananas in the world. There is also a large domestic market for the crop. It's grown mainly in 3 provinces. And worldwide, a major limiting factor to banana production is a virus called banana bunchy top virus. So Dr. Gubba's research focused on this virus. And these are the symptoms that you see on infected plants. As the name implies, bunchy top. The leaves are forming a bunch at the top. And if they get to fruition, they produce very small fruit, which is not marketable. So key findings from Dr. Gubba's work, the surveys demonstrated that the virus is only confined to the South Coast, which is a good thing. It has not yet spread to the other big producers like Mpumalanga and Limpopo. So efforts to contain the spread of the virus on the South Coast need to be continued through active participation of stakeholders. There's need for continuous monitoring through scouting and raising awareness about the virus. And there's also need to critically look at the role of alternate hosts, the weeds. What role do they play in the epidemiology of the virus? And Dr. Gubba published part of her findings in Archives of Virology. And then remember the maize lethal necrosis disease that I mentioned at the beginning. At the moment, this disease is prevalent in East Africa. So we had a student from Kenya, Dr. James Karanja. So he was working with Kenyan material and he was able to select 4 promising lines with tolerance to the disease. There is need to test these lines across maize agroecological zones in Kenya. And there is one of the publications from Dr. Karanja's work is shown on the slide. So now, if you thought viruses were small, now we are even going smaller. There are a group of pathogens called viroids. Viroids are tiny, they're naked nucleic acids and secular, and they cause havoc in the crop industry, especially in the avocado industry in South Africa. And this was the work of Dr. Zwane. So avocado farming in South Africa is a multi-million-dollar industry that caters to both the domestic and export markets. So the avocado sun blotch viroid is a major threat to this industry as it makes infected fruit unmarketable. As you can see from the slide here, these are infected fruits with scars. Can you imagine if these were on display at a supermarket? I don't think any one of you would give them a second look. So if you're a farmer and you're exporting, these serious losses. So there's need to develop sensitive tests to detect the viroid in symptomless plants. Because with this virus, sometimes the plants don't show that they are sick. They don't show any symptoms. So we need sensitive tests for picking up the viroid. So Dr. Zwane's research sought to address some of these issues. And their key findings, she demonstrated that trees with medium and high viroid concentration produced less excessively. They flowered excessively, but lost their leaves during the flowering period. Obviously, this was going to have an impact on their yield. And it was demonstrated that yield comes, compared to healthy plants, you'd get 83 to 96% lower yields. And post-harvest studies also showed that fruit from medium and highly infected trees significantly lost firmness and colored up more rapidly than fruit from healthy plants. This is not good if you're in the export industry. You're exporting your plants, and before they get to their destination, they're already soft. So there will be a loss to you. So conclusion here, flowering, bearing with shedding of leaves and lower yields can be used as indicators of the presence of the viroid in the orchards, okay? But we still need to do more work on the testing, which has to be very sensitive. As you can see from this publication, 2023, Dr. Zwane is a newly minted PhD. So it is the first publication in the European Journal of Plant Pathology. There's a journal called Plant Disease. It is a section in which it allows scientists from around the globe to publish first reports of any pathogens that they discover. So you're discovering it for the first time in your country. You make a report and you publish in this journal. So our research has contributed significantly to this section of the journal, where we have published a number of first reports of viruses we have discovered on different crops and different wheat species. And this is just a list of some of the publications that were put in through to plant disease. So now, moving on to future perspectives. So with viruses, you have to be on top of the game all the time. Remember, I told you they are like a moving target. You can never pin them down. So you can't rely only on the conventional strategies. Biotechnology has helped a bit, but now there's a new kid on the block. Now we're talking about gene editing using what is known as CRISPR-Cas9 technology. So this CRISPR-Cas9 technology is, as I said, it's a new arsenal to control plant virus diseases. It is a gene editing technology that has wide application in improving different traits, including virus resistance. In other words, you can use it for many different traits in crops, okay? The technology is based on the biology of bacteria infecting viruses that are called bacteriophages. So it's not all doom and gloom when it comes to viruses. They also have got some beneficial aspects. This is one beneficial aspect of viruses, these viruses that infect, that infect bacteria. And so this is a cartoon of how these bacteriophages look like. They're very beautiful, look like a spaceship, okay? So these bacteria store sequences of infecting viruses between clusters of regularly interspaced palindromic repeats. This is where the CRISPR name acronym comes from. So this is in the genome of the bacteria. You have the pieces of the virus being stored. So when infected by the virus again, the bacteria uses the stored sequence to seek the newly infecting virus and destroy it. It's a form of adapted immunity. So with the help of a nuclease protein, which is called Cas9, Cas stands for CRISPR-associated 9. It acts as a molecular scissor, okay? This technology has now been adapted to edit genes of choice. So in the lab, this is how you utilize the technology. You create a synthetic CRISPR array that targets a specific gene in a DNA sequence. You then attach a Cas9 cutting enzyme to that array, and then the modified CRISPR array then seeks out the target sequence. The Cas9 then cuts into the gene. Once the gene is cut, it becomes possible to add or delete sections of the targeted gene, thus altering its function. In fact, what I've just said is summarized in this slide. In yellow is what you can synthesize in the lab. In blue is the target sequence that you want to cut. So if you attach the brown, which is your Cas9 enzyme, to the yellow, which is what you've designed in the lab, you can then use it. It seeks a similar sequence, and then when it hybridizes, then the enzyme will come, the Cas9 will come and cut it. So that's how you edit the gene. So once you cut it, you actually are altering function. So against that background, I was privileged enough to be a Fulbright visiting research scholar in the beginning of 22, which that happened to be my second sabbatical, where I was attached to the USDA Department of Agriculture in the vegetable laboratory, also working with Clemson University in South Carolina, Charleston. So here, I was focusing on using CRISPR-Cas9 technology to edit a gene in sweet potato. The idea was to work on that sweet potato virus disease. Remember, the one that results in 80 to 90% yield reduction. So using this technology, you can manage that disease. So I was doing this in the lab of Dr. Kailing. Me and Dr. Kailing go a long way. We were in the same lab during our Cornell time. So importantly, when you develop plants this way, they are not considered to be GMOs. We know that GMOs are controversial. So if there are no GMOs, it means there will be no regulatory hurdles that will speed up. This will speed up the process of commercialization. This technology lays the foundation for the next phase of our research. While I was on my sabbatical, I also worked on weed host of vegetable viruses working with Dr. Matt Kutile of Clemson University, and I presented the results of this work at the American Society of Horticultural Science meeting in Chicago. And this is me here interacting with South Carolina vegetable farmers, telling them about the work that we're doing, why it's important to them, a part of community engagement, if you like. While I was in the US, one thing that was taking root was this controlled environmental agriculture. I was taken by it. What they are doing here is taking disused shipping containers and taking them and changing them into vegetable farms. So this is some form of hydroponics agriculture. You simply plant your crops, the nutrients are there, the lighting is right, you close the doors and come back to harvest. This was amazing. I think it's technology that could find its way to Africa anytime soon. My motto when I work is to find the right balance between work and leisure. So as we all know, all work, no play made Jack a dull boy. So I make sure I'm not a dull boy. I find time to play golf so that I can relax. In conclusion, the future looks bright for my research program. Current research is just the tip of the iceberg. Going forward, there will be more focus on using the CRISPR-Cas9 technology as a tool to manage virus diseases. We'll also look at using artificial intelligence and fourth-generation technologies to predict outbreaks of insect viral vectors as a management tool for virus disease control. And when it comes to academics, I think this slide sums it all. This is what it's all about. Graduating students, putting smiles on their faces and their families, but most importantly, our contribution to human capital development. I would like to acknowledge a few people, students who did the work, the many honors students, master students who I didn't mention by name due to time limitations. Your contribution to my journey is much appreciated. The co-supervisors, I would not have done it alone. My PhD supervisor, Dennis Gonzalez, for his invaluable advice. My colleagues in the discipline of plant pathology for a collegial working environment. I have numerous friendships I've developed over the years. Thank you for all your support. UKZN for giving me the opportunity and platform to follow my dreams of teaching and doing research on plant viruses. And the funders, my extended family members in all corners of the globe. And lastly, to my wife, Mpo, for your steadfast support and the love over the years, for being part of the journey right from the start. My son, Tondera, and my daughter, Rutendo, for the love and for keeping me grounded. Thank you for listening.
Thank you, Professor Gubba, for that very interesting and inspiring lecture. You have certainly contributed to the field of crop sciences. Viruses are definitely dynamite in small packages. Thank you for your service to the community and the broader scientific community. You have made your presence felt in both the discipline and added to the body of knowledge of science. Thank you to the family and friends who are here today and your colleagues for attending today's lecture. My apologies for the initial technical glitches that we had, but I'm sure that you'll agree with me that it was well worth the wait. So thank you once again, everyone, and I wish you all a pleasant afternoon. Bye-bye.